Neha, have you got the YouTube link?
I'm already on YouTube.
I shared the YouTube link here also, Zoom.
Yes.
Okay. Now I'm going to share it in Facebook and LinkedIn. Okay, sir. Now we can start. Yes. Give me 1 minute. Liberty, okay?
Yeah.
Okay.
You ready?
Okay, ready. I'm just going to start. Welcome all of you to our Bioingene live webinar series. This is our 12th webinar in this series. Liberty from University of California, Davis will introduce our speaker, Dr. Manoj Prasad. Neha will collect all your questions from the YouTube chat box. Shoma will join us for interview of the speaker. Today, we're very happy and very glad that our speaker, Dr. Manoj Prasad, allow us to host this webinar and accept our invitation to this platform. I request to Liberty, please introduce our speakers.
Sure. Thank you, Shiva. Welcome, everyone, to today's webinar organized by Bioingene. Bioingene is a nonprofit organization created to promote plant science research worldwide. The webinar series has been designed to build a platform from where plant scientists can present their research to the world. We hope that future scientists can gain perspective and inspiration by listening to the esteemed plant researchers talk about their scientific accomplishments and their thoughts on the future of plant science. We are grateful that many renowned scientists have accepted our invitation to share their research insights with us. We have received a huge response of more than 2,500 registrations for today's webinar. We welcome the audience and thank them for their interest in Bioingene. It is our request to all viewers to please hold all questions for the question and answer round at the end of the presentation. You can type in your questions related to today's talk in the YouTube comment box. You can apply for a certificate of participation via the feedback link and password, which will be provided after the presentation. Submission of the feedback form is mandatory in order to receive the certificate. The topic for today's webinar is Mainstreaming Underutilized Crops, the Story of Foxtail Millet by Dr. Manoj Prasad. Dr. Prasad is working as a senior scientist, professor, and J.C. Bose National Fellow at the National Institute of Plant Genomic Research, NIPGR, in New Delhi, India. He received his PhD degree from the Department of Botany in the University of Calcutta in 1999. Prior to working at NIPGR, Dr. Prasad served as an Alexander von Humboldt Fellow from 2000 to 2003 at IPK in Gatersleben, Germany. His research interests include plant molecular genetics, genomics, and stress biology. Dr. Prasad has published more than 140 research papers, reviews, and book chapters in reputable journals. He is also serving as an associate editor in renowned international journals of high repute, Scientific Reports, BMC Plant Biology, +1, Plant Cell Reports, Functional and Integrative Genomics, Plant Molecular Biology Reporter, Plant Breeding, Journals of Genetics, etc. He is the recipient of many prestigious awards like the Alexander von Humboldt Fellow in Bonn, Germany, the Young Scientist Award, the NAAS Associateship of NAAS, New Delhi, India, the H.C. Memorial Award of the Indian Science Congress Association, the National Bioscience Award of the Department of Biotechnology from the Government of India, the Tata Innovation Fellow of the Department of Biotechnology, Government of India, the J.C. Bose National Fellow of the Department of Science and Technology, Government of India, the Golden Peacock Award of the International Buckwheat Research Association, Slovenia, and the Fellow of 3 national academies, INSA, New Delhi, NASI, Allahabad, and NAAS, New Delhi. We are truly honored to have him with us today. Thank you, sir, so much for being here. I would now like to request that you share your screen and begin your talk whenever you're ready.
Thank you, Liberty, for introduction. Sir, you can again share your screen.
Okay. You can see this?
Yes, sir.
Yeah?
Fine. Yes.
Okay.
Yes, it's okay, and it's already full screen. Okay, sir.
So, good morning, everyone. Thank you, Kelvin, for my introduction. And first of all, I would like to thank the organizer, especially Subhabroto, for this invitation in the webinar series. And as you know, the nation or the entire world is facing such a difficult situation due to the pandemic spread. But we proved that our academia is unstoppable, and we keep organizing such online programs to keep the scientific community engaged in active research and providing research updates. I appreciate the participants also who are gathered in large number to prove that nothing can stop us from learning, and I hope this webinar series provides you an updated information on the roadmap for outcome-based education and research. Today, my topic has relevance to the ongoing issue of hunger and malnutrition. However, I would be talking more on the genomics part of crop improvement with Foxtail millet as an example. I am moving further onto the topic that is here. You can see mainstreaming underutilized crops, the story of Foxtail millet. So, as you know, the humans are the last one to evolve on this earth, but we are leading the game in terms of numbers. As of yesterday, this time our population count was 7.7 billion, and expert predicts that it will reach 10 billion by the year 2020. To this population, we have assigned three basic things as their basic need, namely food, shelter, and clothes. We have been told that without these three needs, survival of the human is not easy. But if you ask whether all the 7.7 billion gets the three basic needs, then the answer is no. 821 million are still hungry. For every 10 seconds, a child dies from hunger-related causes, and 1 in 4 of the world's children suffer from malnutrition, which altogether means that getting food alone will not serve the purpose, but getting nutritious food will. If you see the Global Hunger Index data of 2019, the Africas and South Asia, including India, are still in the danger zone of hunger, irrespective of several steps being taken, including the Green Revolution. Adding to this, the global report of food crisis for the year 2020 shows that 135 million people living in 55 different countries are facing acute food insecurity. This report was released before the incidence of COVID-19, and it is expected that the number would have been now increased due to several reasons, including lack of income and economic constraint among the vulnerable population. This slide shows the statistics on the effect of food insecurity among children. The Joint Malnutrition estimate that 144 million children under the age of 5 years are stunted. 47 million are too thin for their height, and 38 million children are overweight. If you see the percentage of stunted children, South Asia has more than 30% of the stunted children, that amount to 55.9 million children. In case of wasted or too thin children, again, we talk with 14.3%, which amount of 25.2 million children. So realizing the grave situation and to build a better world for people at all levels by 2030, the United Nations formulated 17 sustainable goals, among which 3 are more directly related to food. This underlines the severity of the issues we are talking about. When it comes to food, I want to show an important issue. The largest and the most diverse botanical garden in Kew estimated that there are 11 lakh vascular plants. Among these, 7,000 were used as a food by the early humans. which in due course of time shrunk to just 3 crops, namely rice, wheat, and maize. These 2 crops cater more than 50% of world food. In addition, there are 12 more crops or plants that altogether 5 animal species supplement 75% of the world's food. Now you may understand the question of how to feed 7.7 billion with some 15 species. That is the reason why we say that there is a high demand or time to focus on mainstreaming the underutilized crops for food and nutritional security and using the next generation genomics tools or for the trait improvement in this crop. So if the participants needs more information on what I am speaking about, then they may refer to a recent article published in Advances in Genetics last year. Coming to the first part of my talk, that is the mainstreaming the underutilized crops for food and nutritional security. We call the crops as underutilized when they are cultivated in certain region and consumed by marginal population. These species could be tubers, legumes, leaf vegetables, and minor millets. In case of tuber, it could be taro, arrowroot, Indian shoot, granola, purple yam, air potato, elephant foot. In case of legumes, it may be wing bean, jack bean, bambara groundnut, kidney beans, fava bean, lima bean, cowpea, horse gram, rice bean, velvet bean, ajusi bean. In case of small or minor millet, it could be finger millet, foxtail millet, proso millet, little millet, banyan millet, teff, kodo millet, fonio, job steer, guinea grass, and brown top millet. So India is the second largest producer of millet next to China, and area-wise Rajasthan and Maharashtra are the toppers. But it seems that the production-wise Tamil Nadu, Uttarakhand, and Andhra Pradesh are leading the race. If you see the nutritional potential of millets, it clearly shows that millets are rich in protein, fat, ash, etc., as compared to rice and wheat. In this table, you can see or highlighted in blue color all this composition in 100 gram of grain for all the millets as compared to rice and wheat. In case of micronutrients, including potassium, calcium, phosphorus, magnesium, sulfur, zinc, iron, boron, manganese, and nickel, millets are proven superior than major cereals. Similarly, like the other the previous slide. So if you want to read more about the nutraceutical potential of millet, please read our paper published in Plant Science. So millets are slowly growing, gaining popularity through, as you can see, the media now speaking about it. Professor Bandirajan from Tamil Nadu Agriculture University, Madurai campus, who has released a banyan millet variety, MDU1, for cultivation and consumption that was overtaken the cultivation of rice in Madurai district of India. So recognizing the potential of nutritional potential of millets, our central government has declared all the millets as nutraceuticals, and the United Nations had declared the year 2023 as the International Year of Millets. But still it is miles to go to mainstreaming millets in research and to the plates of everyone people. So coming to the second part of my talk, that I will show how genomics tools can help us to working out one of the minor millet called foxtail millet. So the foxtail millet is similar to rice genome, but maybe it is worked out that it is smaller than rice genome. It's a diploid, haploid chromosome number is 9, a short life cycle, genetically close to major cereals and bioenergy grasses, and it possesses high degree of adaptation to dry environment. That's why these millets are grown in the semi-arid region. And the most important part of this millet is this, that they have better water use efficiency and nitrogen use efficiency, and they are highly nutritive, as I showed. So if you see this graph, that to grow 1 gram of dry biomass of maize and wheat, you required almost 500 liters of water. And but for foxtail millet or other millet, it is just half the water required for producing millet. So for any crop improvement program, you need a good number of germplasm collection, and then you have to screen this germplasm so that you can find out the diverse population, and also screening should be perfect. So this graph shows that the map shows the drought prone areas from where this foxtail millet accession has been collected, and in addition to this, we also collected from different parts of the world, that is USA, China, Kenya, USSR, now it is Russia, Bangladesh, Ethiopia, Pakistan, Turkey. So we have 170 foxtail millet accession or germplasm that are collected from different parts of India or drought prone area, as well as from different parts of the world. So to screen for dehydration and salinity tolerance of this 170 foxtail millet accession, we analyze the relative dehydration and the salinity tolerance on the basis of relative water content, lipid peroxidation, electrolytic leakage, and antioxidative enzyme like APX, catalase, and glutathione reductase. This graph shows that we have categorized the 170 accessions into highly tolerant, sensitive, and highly sensitive, and they are grouped here. So by this way, we have identified the most contrasting tolerant and sensitive. So Prasad and IC403579 was highly tolerant with both the stress, drought and salinity, whereas Lepashi and IC480117, they are highly susceptible. So this physiological and biochemical data was further confirmed by the expression analysis of these genes like APS, catalase, and glutathione reductase. So based on this, when we started this work at the 2008-9, at that time NGS technology was not there. So we, based on this cDNA subtractive library concept, we have prepared a contrast, as I showed you, that we have identified four actually contrasting genotype or accession of foxtail millet for both the stress, dehydration, and salinity. And we did the comparative transcriptome analysis, and we found that this is the dehydration and salinity, 18 genes which are common between these dehydration and salinity stress tolerance. And total transcript of both the library consists of 182. So you can see here only 18 genes, that is out of 182, with the 10% genes are common between the two stress when dehydration and salinity was imposed while using the same contrasting genotype. So we suggest a distinct mechanism to perceive and respond for salt and dehydration stress condition in foxtail millet. So then I am coming to a story recently developed by one of my PhD students, Dr. Roshan Kumar. Now he successfully defended his thesis on heat shock protein. And so the title is Identification and Characterization of Stress Responsive Gene Heat Shock Protein as an example. So in the year 2012, the American group of Benedzen and Chinese BGI, Zhang et al., they have sequenced this foxtail millet genome, and this is the description. I'm not going details about the genome sequencing, but how we can utilize this genome sequence information to find out the candidate gene that I want to show you. So from the whole genome sequencing and annotation, we identified that when we compare millet, sorghum, and maize, that 1517 genes, they are millet specific. And out of these 1517 genes, we found through geo annotation, we identified 586 genes were functionally annotated as responsive to water stress. So through this, we pinpointed several candidate genes. One is the heat shock protein, aquaporine, NIP transcription factor, Werke transcription factor, and ADP-RF. So we did the cloning and then the characterization. So coming to the story of heat shock protein as an example, as we know, there are different kinds of heat shock protein, like heat shock protein 40, 90, 100, small heat shock protein. So these heat shock proteins regulate different aspects of stress defense through protein-protein interaction of the target protein. So when we searched the literature, we found that in different classes of HSPs are present, but there is no information available for Foxtail millet. So through this HMM profiling and database search, we identified that 20 members for HSP 100, 9 for HSP 90, 27 for HSP 70, 20 for HSP 60, and 37 for small heat shock protein. So we check the expression profiled by the qRT-PCR in different abiotic stresses like heat, drought, cold, and salinity stress. And for that, the same for two contrasting genotypes, which was used for the cDNA substructive live reconstruction, the Prasad and Leprashi was used as a contrasting parent for this qRT-PCR analysis. And out of this 37 gene of 29 expressed differentially in either one or multiple abiotic stress. So you can see here that only one, that is the HSP27, was expressed differentially in all the four stress like dehydration, heat, salinity, and cold. So this we choose as a candidate gene for further analysis. When we check the HSP27 in different stresses like heat, cold, salinity, and dehydration, we found that this gene HSP27 was almost 35-fold upregulated in heat stress, almost 21-fold upregulated in cold stress, and almost 48% upregulated in heat stress and salinity stress. So to check the abiotic stress tolerance, we use the yeast system to just confirm whether this gene is working in the yeast system or not. So we clone this gene in this vector PYS2, and this was confirmed by this restriction gyration. And then it is the 642 base pair, and then we overexpress this gene in yeast. You can see here that transgenic cells overexpressing this HSP27 showed enhanced tolerance to heat, dehydration, and NaCl 2.5 molar NaCl. So you can see the overexpressing yeast cell of HSP27. They are surviving very well when this wild type is dying. So it shows that in heterologous system, that gene from Foxtail millet is working fine in the yeast. But this is not the conclusive proof, because this is just to show that whether it is working in other system or not. To do this, we overexpress this HSP27 in rice, and transgenic rice lines overexpressing HSP27 was generated, and the evaluation of these positive clones was done in T2 generation. So this is the construct. I'm not going details about the construct, but the result I can show you. This is the Gus expression analysis of the leaves of the transgenic rice plant. And that A to F is the line 1 to 6 with Gus expression, where G is the control, non-transformed leaf, and H is the leaf of transgenic vector control. So it shows that yes, that the lines has been transformed very well. And this is the PCR amplification of HPT2 gene from transgenic rice callus. So when we did the phenotypic evaluation of evaluation of the HSP27 in the overexpressing rice lines, we found that most of the agronomic traits such as plant height, number of tiller per plant, number of panicle per plant, panicle length, number of grain per panicle are observed to be higher in transgenic plant as compared to the wild type. You can see here in wild type and then the three transgenic line L1, L4, L10. So in all the traits, it was improving, but there is no change in weight per 100 grain and length per 10 seeds. So then we did the response of this in ROS, and we found that the transgenic rinds exhibit low ROS accumulation in response to heat stress. And this was also confirmed by the chlorophyll content, and this is the chlorophyll A, this is chlorophyll B, and this is the total chlorophyll content of wild type and the 3 transgenic lines. So the different colors, this orange color is control, and this yellow is the heat the day 4, and green one is the heat stress in day 8. So you can see, if we can concentrate on this last picture, the total chlorophyll content, you can see here the sharp decrease in chlorophyll content was found in the wild type, and in response to heat, about 75% chlorophyll loss was observed in the wild type after 8 days of continuous heat stress, and chlorophyll loss during the heat stress is comparatively much less in the transgenic lines. You can see here the transgenic lines are behaving better, all the 3 transgenic lines, as compared to the wild type. And if you consider this green one, that heat stress at 8 day, so it is, you can see here that it is here. So about 75 to 80% chlorophyll loss was observed in the wild type. So we also overexpress and edit this gene in Foxtail millet, and this was also standardized, and we recently published and showed that almost 27% transformation efficiency was obtained while doing the transformation of Foxtail millet. So this gene was also overexpressed in susceptible cultivar of Foxtail millet. At the same time, it was also silenced in the tolerant cultivar through CRISPR-Cas. So coming to the bigger picture, that how to identify the candidate gene, because as I showed in hits of protein, 113 hits of factors protein was there, but we identified one based on the the expression study. So similarly, for other gene families, we also did this is the pipeline for this how to integrate the pipeline, how to identify candidate gene. So you have to identify the gene family members first, then you do the gene structure, chromosome location, duplication, divergence. These are all in silico. Then the domain architecture, phylogeny, physiochemical properties, promoter analysis, gene ontology, expression profiling. This is all in silico. And then once you identify this candidate gene, and then you do this overexpression in heterologous sequence, like I have shown for yeast, to get an first-hand idea how it works. So by this way, we have identified 124 C2S2 type gene finger protein, 209 MIP transcription factors, 110 genes for Werke transcription factor, 160 genes for the secondary cell wall related C4 biofuel model Setaria italica, and then 23 and 25 genes identified in Alexander italica of ADP ribosylation factor. So a recent one is this one recently published in Journal of Biotech, genomic dissection on expression analysis of stress responsive gene in C4 panicoid model Setaria italica and Setaria viridis, where we have checked all the genes of the stress responsive in both these progenitor, the wild relative of Foxtail millet, that is Setaria italica versus Setaria viridis. So for this is the integrated pipeline for transcription factor studies, and by this way we have identified different classes of transcription factor, and the number written in bracket is the number of genes for the particular transcription factors. And we have developed a database for Foxtail millet transcription factor database. So this is the backbone of this database, and this is the data available in the database. So you can see here that this is the snapshot of the Foxtail millet transcription factor database. So coming to the large-scale market development based on the 2012 genome sequence information of Foxtail millet. So we use the transcriptome data, and then we identify the coding sequence and the intonic region. From intonic region, we have identified the internal and polymorphic markers. More than 5,000 ILP markers have been developed for this coding sequence by using the MISA pipeline. This is a bioinformatics tool to identify the microscopic markers. We identified 500 SSR markers, and this genomic resources have been utilized for genotyping assay. Similarly, for the transposable element-based marker, we have developed 20,000 repeat junction-based marker. and which was used for the genotyping assay. And there are different classes that you can see here, LTR finder like Copia, Gypsy, non-LTR like line sign, and the class 2 DNA transposable also repeat markers, a different class. So there are 4 different types of markers from the transposable element has been developed. So the number consists of more than 20,000 transposable element-based marker has been developed in Foxtail millet. And at the same time, we have also developed a database for that, so that the general public they can use it and they can use in different crops also. So these are the four basically types of marker we have developed. One is the ESSR marker, internal and polymorphic marker, microRNA-based marker. This is the first kind we have developed a microRNA-based marker, and after that people have been developing rice also. And then the transposable element-based marker. So this is the number of markers developed for different classes. This is the polymorphism in Foxtail millet, and this is very important that you can see there that that ESSR and ILP, they are more than 85% are transferable. And for microRNA, it is 50%, and transposable element we have not studied. But the result is encouraging, so that the people working in different crops, they can utilize this information for their crop improvement program. So based on that, we have done this comparative mapping of maize, sorghum, and brachypodium, and these are the stories we have published in 4-5 years back. So coming to the utilization of this as markers developed for Foxtail millet, we did association mapping for the yield traits. And for that, as I said, we have that 170, we actually increases the number, 184 Setaria accessions from diverse geographical regions were phenotype for 10 yield contributing agronomic traits. And these are the traits based on flowering plant height, tiller number, flag leaf, and 1000 grain weight. And for the 3 consecutive years at NIPGR field, and we have identified 8 markers which are associated with 9 different agronomic traits with a statistical value of 0.05 and contributing to 6 to 25% of phenotypic variation. So, for example, grain weight, this microselect marker B260 located in chromosome 1 explain 25% of the phenotypic variation. It means if the trait is governed by 100%, so this marker itself, B260 on chromosome 1, is tightly linked and highly associated with grain weight, and it explains 1/4 of the total variation for the trait of interest. So these are the publications from the marker development, more than 5,000 internal marker, more than and the database. And then this is the ESSR marker development. This is the population structure association mapping, just I showed you. And then when this NGS business comes, then we shift from our generated marker to this NGS thing with the same set of genotypes, the same phenotypic data. We use this NGS technology, and we identified 30,000 SNPs in distributed in 25.9 SNP per MB. Maximum SNP density was observed on chromosome 8, and minimum was on chromosome 9. And these are the distribution of SNP in different chromosome of 9 chromosome of Foxtail millet. And this is the technique how the NGS has used the dehyd sequencing. So this is the cut short of this mapping of quantitative loci for the agronomic traits, and we identified 81 markers which are associated with involving 79 SNP were identified for the different agronomic traits. We also identify some candidate genes, and these are the SNP, and this is the chromosome location of this SNP, and this is the position of the investor in the genome, and this is the SNP variation, and what are the favorable allele, p-value, and the putative candidate genes function. So these are the flag leaf weight, grain yield, 1000 grain weight. So these are the 3 important traits. We have identified putative candidate genes for that. Similarly, we did the same dehyd sequencing for the 10 micronutrients, as I showed you in my introductory slide, that we have also estimated phenotypically the sodium, potassium, calcium, magnesium, sulfur, manganese, boron, nickel, all the micronutrients, the 10 micronutrients we have estimated in the whole population, and we identified 74 marker trait association for the 10 micronutrients. So this table shows the candidate gene we identified for both all the micronutrients, boron, magnesium, zinc, and iron. And these are the chromosome number, and these are the SNP position, and this is the desirable allele linked with this high boron thing, and these are the candidate genes. Last year we published this thing in Journal of Cereal Science. So whatever we have developed with the genomic resources, we have basically put it in a form of a web resource, and there are 4 different web browsers we have developed. And in Foxtail Millet, this is one of these kinds, the Foxtail Millet Marker Database, a versatile database of Foxtail Millet Marker for millets and bioenergy grass research. So if I cut the story in one slide. So when we started this story, then at that time only 26 TSSR and 98 ILP marker was available in the literature. For the gene, DEB, NAC, and WD40 was partially characterized, but over the time period, you can see here we have developed thousands of different kinds of markers in Foxtail millet. Similarly, we have characterized several gene transcription factor family members in Foxtail millet. So these are the databases: the Foxtail millet marker database, Foxtail millet transposable element marker database, and this is the Foxtail millet transcription factor database. So anybody can go, and there is a very good web service was there, comparative mapping, CMAP, so that you can compare our Foxtail millet resources with other imported cereal stock also. So if you want to know further about what we have done in Foxtail millet, so you can read our this review article and published in 2015, Theoretical and Applied Genetics, Advances in Cereal Genomics for Genetic Improvement of Cereal and Bioenergy Grasses. And then we also published a dedicated book to Foxtail millet genome. And last year in August, in this month, we have conducted one-day symposium that's neglected and underutilized crop species for food, nutrition, energy, and environment at NIPGR with the thematic area of genetics and genomics of orphan crops. All the orphan crops I showed in the introductory slide, they have been not only millet, other tubers, legumes that had been presented in this one-day symposium. And then the important session was also conducted, tapping the wild germplasm for crop improvement. This is very important that in the current scenario. So this is in nutshell about the Foxtail millet story. And I can thank my students for this. And the HSP work was done by Roshan Kumar Singh. Last month he defended his thesis. Now he become doctor. And the database and the genome study has done by Muthu. He is now faculty. I think last time he has talked in this webinar series also. So Muthu has developed all the databases and the genome study from Foxtail millet. And the work has, whatever I presented today, was mainly funded by Department of Biotechnology, Government of India, and partially by Department of Science and Technology. So thank you very much for your patience hearing, and I will answer the question with what I have got from the different participants. So this is all over from my side, Subhabroto.
Yes, sir. Sir, we we collected few questions from YouTube, and I pasted here in the chat box. Just here is the name and the questions of the.
So this is the first question. If I remember correctly, what role will Foxtail play in global food security?
No, sir. It is we are coming on that. Okay. These are the. Here we are going to paste the questions. Just a minute, sir. This is the first question, sir, from the Orunima Banerjee.
Hmm.
Uh, can you get this question? This is the chat box there
Chat
on
box, huh?
the blue just beside the blue skin sharing option in the bottom of the your screen
Chat. I got one.
chat.
Yes, yes.
So
Thank you, Professor Prasad. It was a pleasure listening to your talk. It was very late for me, so I must.
it is the liberty. Ignore that one. This one.
How can we reduce goitrogens in millet? Arunima Banerjee University, bird one. Goitrogens, I have no idea about these goitrogens.
No problem, sir. There is I just give you more question. You choose from there any one, two, three
But
questions.
I will definitely respond to goitrogens because I have to also look for it.
Okay.
So the second question I have seen here from Sundarasan, what type of sequencing method is adopted and whether de novo assembly was executed to compare the transcriptome. The thing is this, the 2012 paper, when the first genome sequencing was done, that was de novo sequencing, and now we don't need to do de novo because already a gold standard genome sequence is already available. So we do resequencing with a very cheaper price so that you can just map the reads on the already sequenced information. The next question is how osmotic effect of salinity reduce plant water uptake? This is physiological question. Next one is there any hidden medicinal potential in foxtail millet? This needs to be addressed because there is no publication, at least with justification about the medicinal potency. But I hope it should be because nutritionally and in other traits it is highly efficient. It also should be looked out. Is there any effect on human being on transgenic millet? There is no information on transgenic millet because we are developing for the first time and it should be tested in different RCGM protocol of Department of Biotechnology. So until unless we test, we cannot say. But in general, if I say from other crops, till date, there is no negative effect has been reported for any transgenic crops. So the next question, quality parameters and storage potential of millet. Is there any genes for seeds longevity are identified in millet? No, this information is very important, but we are also working on it for seed longevity. Uh, Yes, this is very important. So, Sanju from I don't know from where. Surjit Sanju. He asked that whether foxtail millet can be used in diabetic patient. Yes, that is the characteristics of all the millets that their glycemic index is very low and they contain good resistant starch. So the glycemic index become very low. So what you do when you eat the carbohydrate uh, and due to presence of good resistant starch in all the millet crops, the conversion of carbohydrate to glucose is slow. So the transcription of glucose in the blood is become slow. So all the millets, due to presence of good resistant starch, they can be a good use for the type 2 diabetic people. Can foxtail millet be a substitute for rice consumed on daily basis? Yes. If you go in the southern part, now you will get ragi dosa and everything because nutritionally this crop is 5 to 6 times better than rice and wheat. And then in top of this, there no need of taking proper care of these crops because they grow in marginal land where there is no need of fertilizer, no need of water. So nutritionally better 5 to 6 times, very less water is required. So I think millet is the substitute for all the major crops. So today's lecture I have given to show that how important the millets are as compared to the rice and wheat. Which is the better consumed for oats and millet? They are all same, but millet will be better. Joseph, so my question is, when it comes to identification and overexpression of genes, there are many genes involved. Why did you focus on working genes? That is the reason I told you that the story of hits of protein. 113 hits of factors genes have been identified. We cannot characterize 113. So that is the reason we developed this pipeline, bioinformatics, through HMM profile and bioinformatics tool to identify the different classes. Like you asked me for the worky genes. For worky genes, we have identified more than 100, but we cannot characterize 100, more than 100. So we test these genes for different stress, and then we find out the potential candidate which has been showing differential expression in most of the stress. So like in HSP, I have showed this HSP27 showed differential expression in all the four stress like heat, drought, salinity, and and cold. So this is the potential candidate. How we can pimp out the uh, number one or two or three genes. Sir, is there any study on reported to qualitative traits in foxtail millet? No, we are doing all quantitative traits, but I think there should be, because if I think TNU, some Ravi Keshavan and Bandi Rajan, they are doing the qualitative traits in foxtail millet, but not only foxtail millet, they also work on podo millet and banyan millet. Anti-nutrients such as phytic acid and tannin present in this tuna. This is a very good question by Prabhat Kumar Singh. Anti-nutrients such as phytic acid and tannin present in this millet, can we improve this using genomic approach? Yes, and any improvement in this area is going on. We are working on it, and my student Muthu has an ECRA project on phytic acid in foxtail millet and how to reduce this phytic acid content in foxtail millet. He's working on it, and we identified one candidate gene, and we try to actually silence this gene so that the level of phytic acid we can control in foxtail millet, because this is the problem for rancidity. So if you keep this millets for longer duration, it tastes better, bitter. So how do select favorably regarding the trait of interest in GWAS? Because that is the reason GWAS will give you an overall idea. But with this, when you try to find out the candidate gene for the most tagged one, most linked one, then we have to get an idea that what are the candidate genes that has been identified through genomic approach by through GWAS. What are the necessary steps taken to commercialize the popular of foxtail among people in the world? So you can see that if you go to the market, in supermarket, you will get all the millets, but their price is more than rice and wheat because the production is less, and the people for other countries, but mostly the European and American, they are liking millets due to this nutritional property. So that's why in India, the cultivation is less, production is less. That's why the whatever production we have, the price is very high because demand is there. So as I showed you one story about this Bandi Rajan, Professor Bandi Rajan from TNU Madurai campus, he developed a hybrid barnyard millet that has replaced the rice and that in that region. So by this way, we can commercialize and popular foxtail millet from by showing good improved quality of millets in both in terms of production and quantity. Sir, can the toxin in millet can be reduced by identifying those genes in manipulating them? If so, how? So as I said, the toxins in millet like tannins and then the phytic acid. Similarly, we have to phenotype the different toxins in the range of germplasm, divert germplasm, and then we have to do genotyping, the GWAS, and then we have to identify the genes, candidate genes, which are linked with these toxins, and then we can identify the candidate gene from based on the GWAS study. How can we such millet get available in Nigeria soon? I think equally said that the CGIR system, they are using a lot of money, investing a lot of money for this millet crop grown in this all this sub-Saharan Africa. So I think this millet is now available in Nigeria and other African countries. They are well grown. Variety is suitable for rainfed moisture stress condition with highest yield. Khushbu Chandra. I think I need to get this information from my Tamil Nadu Agriculture University people because they are the breeder. They can give me the idea, tell me what are the suitable variety for rainfall moisture stress condition with highest yield. So I can get back to this question later on.
Okay. Thank you, sir.
I didn't see any more questions.
No, sir. It's actually we select few one because somebody asking for rice, someone asking for different crop also. So
It's a never ending.
yes, and this is a plant science or plant platform. So many people are try to relate their work with speaker's work. So it's
Yeah,
happened.
yeah.
So now we going for our interview session. Now I invite Soma to
Uh-huh.
interview you to know your journey because many younger students and researchers are there. So we start your interview. I hand it over to Soma.
Hello.
Hello.
So it was it was a wonderful presentation. Thank you so much, sir. We are now moving ahead to the interview section of today's webinar. Today
Uh-huh.
is our 12th webinar of the series. We are honored to have with us a very distinguished and senior scientist, Dr. Manoj Prasad. Sir, you're a pioneer in your field of research. We would love to know how you started your career. Please tell us your journey so far as a plant scientist.
So basically, I was born in India district where our previous Prime Minister, Mr. Chandrasekhar, was belongs, and uh, and I just was born but brought up in Bengal. So my father was an ICR scientist, and he actually came from UP for such a job, and then I was totally educated in Bengali medium, and uh, and I started Bengali speaking better than Hindi. So when I get married, my wife is scolding me that why you have changed the gender, because in Bengali there is no gender, as you know. Jol khabo, water is in Hindi, well, they never eat it, they drink it. So, but that is the thing I accept. But nevertheless, I enjoyed my stay at West Bengal, and I have a soft corner for the West Bengal, and especially for my district Nadia, from where I was brought. And then I did my graduation from Rohra Ramkishan Mission, because I
Okay.
was trying to study the zoology, but I
Okay.
don't get the seat for zoology. Because you know that for a 10 plus 12 student, studying plant is very boring actually at that time. So this is a common thing. This is not hiding. I am also not hiding, but I tried a lot that I should stick to the geology because it is related to human, and then you can correlate most of the things. But after 6 months, I also got a chance. But my Maharaj, that Dibyanand, at that time, he called my father along with me, and then he told that he knows my father. So he told that you are an agricultural person, agriculture scientist, and your child is related to showing interest in geology. Then he gave a very good example. He told, look, if you are studying botany in plant science, then you can move to different branches like environmental, forestry. But in geology, you will be very confined to some specific area. So he convinced me and my father that botany is a very good subject and you should keep doing this. And that was the change in my life. And I stood university second actually on that batch, 92 batch.
Wow.
And then 94, I was admitted at Baligon Science College Botany Department. And then after this, I was fortunate that Professor Arun Sharma and Archana Sharma, they taught us and then I enrolled as a PhD student with Professor Rajesh Chaudhary at Botany,
Okay.
but I was actively involved with Professor Arun Kumar Sharma for my work. And then in 1998, once I submitted my thesis at Calcutta University, I was fortunate to join another legend, Professor P.K. Gupta. Most of you have read
Yes.
his books in
Yes. Yes.
Elements of Biotechnology and Cytogenetics. So in the year 1998, I joined Professor Gupta in wheat biotechnology project. So whatever I knowledge, because in Calcutta University, in West Bengal research at that time, in molecular biology, you are the front runner because we have that Rockefeller Foundation project from USA in rice, and then our lab was most of the Bose Institute and Calcutta University labs are highly actually well infrastructure was well. So we have PCR at that time. Nowhere you will get PCR and other instrument to do molecular biology. So as a molecular biology training, we get a very good training, and we conduct training for the other people parts of the different parts of India, and uh, and and then when I joined Professor Gupta's lab as a research associate for a wheat biotechnology program, and within 2.5 years, I have published more than 10 papers. And that is in tag, 5 of them are tagged. At that time, tag was the Bible for all the breeders and the geneticists. So I got this Humboldt Fellowship after 2 years, and then I moved to IPK Gatersleben, Germany. And there also I worked with, first I worked with wheat with Marian Reuter, and then second, I shift to barley as a postdoc after finishing my Humboldt Fellowship. So I spent 3.5 years in Germany, and then at one day I got a mail from Professor A.K. Sharma, Arun Kumar Sharma, my teacher from Calcutta, that there is one new institute known as National Center for Plant Genome Research at JNU campus, and Professor Ashish Datta is the caretaker. He is looking after it. Then he was at that time he was the vice chancellor of Jawaharlal Nehru University. So if you are interested, you send your CV. I sent my CV, and then one day I got the interview call that I was asked to. At that time, my wife was pregnant, so I told them that this is the case. How could I move? So they told me at that time there is no online things. So that is another thing that in the year 2000 there is no online. So anyway, I told my wife told no, you go and just go and give the interview, and the night flight you can come back. So I did the same way. I came here, and then I appeared in the interview, and then I returned back. So and after 7-10 days, I got the letter, appointment letter. So this is the story in nutshell. In 2004, I joined here. At that time, it was NCPGR at JNU campus. And now it is National Institute, NIPGR. And the whole story starts from 2004 onwards. And what I am today, due to this excellent infrastructure and good scientific environment which is present in this institute. And whosoever is coming as a student or a teacher, they all excel if they have the willpower.
Definitely, sir. So true. Lovely. Your story was amazing. Sir, my next question is that what role will foxtail millet play in global food security?
Okay. So I can talk about that. Foxtail millet, along with other small millets, has a collective role in securing not only food security but multiple security. This includes food, nutrition, fodder, bioenergy, and biofuel. First, the millet were consumed by our ancestors, and it is not new to us. So it has the potential to address food security. Second, millets are 6 to 7-fold nutritionally superior to major cereals, and thus it can address the nutritional security. Third, millets are now majorly used as a feed for cattle. This is
Okay.
for the animal food, and it has been proven to be the best feed for fodder as a fodder crop.
Okay.
Fourth, millets, particularly foxtail millet, are genetically close to biofuel crops like switchgrass, napier grass, etc. And these
Mm-hmm.
millets also possess excellent biofuel traits. So
Mm-hmm.
that is the only reason why the American and the Chinese group have sequenced this crop. And thus,
Hmm.
I strongly believe that harnessing millet would address multiple securities. I hope I can answer their question.
Yes, definitely, sure. Sir, my next question is that what modern techniques
Uh,
should be implemented for future plant science research?
So, modernization is already there in plant science research. Several techniques such as kit-based, and we have now sophisticated instruments to perform complex studies and analysis. So, basic research has no issue with modernization. However, applied research needs the intervention of artificial intelligence, and artificial intelligence-based tools are required for climate prediction, analysis of soil condition, insect pest attacks, more like an early warning system to avoid crop yield, so crop loss. So that's the thing, that if we are moving towards the applied research, then artificial intelligence will play a big role in coming days.
According to you, which sectors of plant science research and agriculture will prosper in the near future?
So in research now, people will look onto integrating genomics. Now the world has realized that focusing on one omics approach, let it be genomics or transcriptomics or metabolomics, etc., will not produce or provide a clear picture. So future research will involve all the holistic approaches to address scientific questions of nation and international importance. In agriculture, as I said, the development of artificial intelligence-based tools will take the stage in the coming years, and we also look forward to such technology breakthrough.
All right. Sir, now we'll conclude our webinar and also the interview with some words of advice from you to the young
Yes,
researchers.
I can give to for the young researchers who are listening. So you people listen very carefully what I'm going to talk about. I always tell three important points to my students. Number one, if you are deserving, you will get what you want. No one can stop your success. So if you are deserving, you will be the winner. Number two, three things. How to think, speak, and write cannot be taught. These
Okay.
skills comes by rigorous reading and practicing.
Hmm.
Only those who masters them will be successful. So three things: think, speak, and write. These have to be developed by yourself. You cannot, nobody can teach you.
Right.
If you can help someone when they are in need of assistance, then you should help, no matter what, without any thinking whether you will get the feedback or not. So you should help. You should be very positive in all the cases. Be positive, you will get everything. My own student who have followed these three are already successful. Murthy is one of them, Charulata is another, and Ashish is now doing, Namisha is also doing. So these are the few examples I can tell you, that if you follow this, practice this rule,
Mhm.
I think nobody can deter you from success. But thing is this, you have to be patient. Don't
Right.
don't utter any bad or harsh
Hmm.
word to anybody.
Hmm. Hmm.
Be positive. Okay.
Yes, sir. The beautiful words of advice, sir. Thank you so much, sir, for being with us today. It was amazing. It was wonderful to have you with us today. Now, let's go back to Shubha. Over to you, Shubha.
Thank you so much.
Thank you, sir.
Thank you, sir. And not only the younger listener, it's also valuable for all of us, including me and our team. This is a very valuable information, and we also believe in these things, and one of the reason maybe the Bioingene platform is created, that
Yes, yes.
we should help others and promote the plant sciences.
Yeah.
Uh, as And I am very fortunate that you and me also share the same campus
Yes,
for
yes.
our PhD, University of Calcutta. Thank you, sir. Today we got so many positive feedback, so many feedbacks. There are so many positive feedback on your talk. So much. Actually, millets is very new. I think many of people very excited about these crops because it has a potential. It has so many resistant genes, and it's just the field is very open, and
Yes.
Lots of work can be done in the future. So the younger students, those, and many of in comment box, many of our listeners and viewers are asking for the topic of their PhD work and MSc work. So I can suggest them as a whole that millets has the potential. There is a
Definitely.
lot of resistant power on it, and genome sequencing is available. So CRISPR can also be used in future very actively on this scale, and is very grateful to listen someone who contributes so many accession in the India and Dr. Manoj Prasad and his lab members, team members are very active in their research field. I know few of them personally, and already one of a student delivered the webinar in this platform. So I thank to you, sir, to give a strength in this platform, and hope in future your work also be we can also host your work to know more successful stories from your lab in this platform in the future. So thank you for joining.
Thank you, Subrata, for all these kind words.
Thank you,
Thank you.
sir. Thank you, sir. It's nice. Sir, we have some few option instruction, and I request you. There is a end button in your the after the mode. You can leave. There is an option that you can
Okay.
leave
Okay.
that meeting. So thank you, sir. Again,
Thank you. Thank you.
uh,
Thank you.
I
Thank
I
you.
send you all the feedback and the listener history,
Yes.
everything in your subsequent mail. Thank you.
Thank you.
Uh,
Thank you.
Thank you, sir.
Thank you.
Thank you, Neha, for helping us. It was a nice webinar, as I can found from the YouTube, and many people are satisfied and successfully. So our next webinar in 11. So we in 11 on August, we again gathered and hosted our 13 webinar in this series. Thank you, Neha. Thank you, Shoma, for assisting us for the uh, uh, interview session, and I I am very much thankful to you for that. Thank you, Shoma. Thanks to all the viewers for for your participation. In our next webinar, I just screen my share my screen. Those who are not getting the password, today's password is 59595. The two upcoming webinar is on 11th and 17th. We arrange intentionally, we reduce the number of the webinar per month. So 2 to 3 webinars in a month we willing to arrange. So our next webinar is on 11 and 17. This is the today's password 59595. This is the feedback link, and this feedback link also available on our website. Go to our website. There is a option of feedback. You can feedback by putting this password. It is mandatory to collect the certificate, as I send all the instruction in the email to the registered participant. Please subscribe our YouTube channel. It's help us to connect with you very shortly, because sometimes any technical problems we can't change the feedback, the link. So I prefer to use a live link which is automatically generated by YouTube. So uh, it can be visible when your subscription is on our channel. And this all from us. Your name and the password will available on our website. Give that give 2 days on that. Within 2 days, there will be a list. Those are the very habituated of our webinars. They can find easily, and those are the new. Go to the bioingene.com. It's our website under construction. There is a link to our alternative website. Go there and collect your certificate. And we try to intimate you, but we can't email you your certificate. You have to collect it from our website. All the certificate, those are giving the successful feedback. All the certificate will available on our website. Please collect it. Don't if you give your feedback, your certificate should be there. So search it and collect from our website. So thanks again, all of you. We'll meet again on 11th August at 11 a.m. Indian Standard Time. There's a talk from Dr. Purvandarkar from Australia. She is doing biotechnology, molecular biotechnology and genomics and different type of work. Very interesting. I heard him several webinars, and I'm fortunate she's accepted our invitation and gave her talk. So we are very excited on her talk on 11th of August. Thanks all of you. I put the screen for a few minutes to ensure that you get the password. And this password and link only shared here. We can give you further via Facebook, via individual mail. It's not shareable after the webinar or after the live session is closed. So I'm sure you all get this link and collect your certificate from our website.